imagine for a moment, just think about this. What if a single treatment, just one time, could permanently get rid of HIV? No more daily pills, none of that lifelong management stuff, just a genetic fix. That's it. It really feels like something out of science fiction, doesn't it? But what if that idea that maybe once seemed impossible is actually, starting to take shape right now in actual labs? I'm really curious to explore this with you to see if this revolutionary idea is becoming a reality.
It really is an extraordinary possibility, and
Yeah.
it fundamentally changes how we've thought about tackling viruses like HIV for decades.
Right.
We're definitely on the edge of something potentially transformative here.
And the implications are just huge, aren't they? Especially for a condition that's always meant lifelong treatment. So that's exactly what we're diving into today. Our mission really is to explore gene therapy's promise for actually eradicating HIV permanently. With our expert guide here, we'll dig into the latest research, try to understand the mechanisms, the breakthroughs, and the big challenges too. We want to figure out, can gene therapy really truly end HIV for good?
And, to really get a handle on the cure, we absolutely have to understand the enemy first. We need to understand HIV itself.
Okay, good point. So let's kick things off there, maybe start broad. What is gene therapy, generally speaking?
Well, in simple terms, it's a pretty revolutionary medical approach. Scientists basically go in and modify or sometimes even replace specific genes inside your body.
To treat or prevent disease, right?
Exactly. And when you apply that concept to HIV, the goals get very specific, very targeted. We're talking about maybe deleting or editing the genes that HIV actually uses to get into your cells.
Blocking the doorway.
Precisely. Or strengthening your own immune system's natural ability to fight the virus, giving it a boost, and also blocking the pathways HIV uses to make copies of itself once it's inside.
So it's more than just treatment then. It's like reprogramming the body at the genetic level to basically outsmart the virus forever.
That's the ultimate goal, yes. And what's really interesting is how HIV works, because it operates at that same genetic level. HIV specifically targets your immune system, mainly these crucial cells called CD4 cells.
Ones that coordinate the immune response?
Exactly, those. And it uses a kind of key, a co-receptor on the cell surface called CCR5, to unlock the door and get inside. Once it's in, it does something quite sneaky. It inserts its own genetic instructions right into your human DNA.
Wow.
Yeah, it essentially hijacks your cell's machinery to just churn out more virus copies. So the big scientific light bulb moment was realizing, wait a minute, if we could block that entry point, that CCR5 keyhole.
Or maybe even remove the virus's instructions from the DNA.
Exactly. If we could do that, we might be able to stop the virus cold. That insight shifted the whole focus from just managing it to thinking about actually eradicating it.
That leads us perfectly to a really pivotal moment, doesn't it? A real-world story that kind of proved this concept wasn't just theory. I'm talking about Timothy Ray Brown, the Berlin patient.
Yes, a landmark case.
His story is incredible. The first person widely considered cure of HIV. He had leukemia, right? And needed a bone marrow transplant.
That's right. And crucially, the bone marrow donor had a rare natural genetic mutation. A mutation in that CCR5 gene we just talked about.
Making them naturally resistant to HIV.
Exactly. So after Timothy Ray Brown received that transplant,
What happened with his HIV?
it became undetectable. And quite remarkably, it stayed undetectable for over 12 years, right up until he sadly passed away from cancer.
12 years. Undetectable.
Yes. In that case, Timothy Ray Brown's experience, it provided the very first clinical proof, real-world evidence that, yes, changing the genetics could actually cure HIV.
It must have sent shockwaves through the research community.
Oh, absolutely. Fundamentally changed the game, showing that eradication wasn't just a dream.
Okay, so that was a monumental step. But a bone marrow transplant like that is incredibly risky, complex, not something you can just roll out widely. How is modern gene therapy building on that initial success? Are there, less drastic ways to achieve a similar outcome now?
That's the key question. And yes, the field has moved significantly beyond that. We're now looking at more targeted, potentially safer approaches. There are broadly three main strategies being explored right now.
Okay, what are they?
First, there's CRISPR gene editing. You've probably heard of CRISPR.
Yeah, the genetic scissors tool.
Pretty much. It allows scientists to go in with remarkable precision and literally snip out the HIV genetic material that's been integrated into an infected cell's DNA. Early trials are looking promising, showing they can reduce HIV levels, hopefully without damaging the healthy parts of the DNA.
Wow, literally cutting it out. Okay, what's the second approach?
The second is directly inspired by the Berlin patient. CCR5 deletion. Instead of a whole bone marrow transplant, scientists use gene therapy tools, sometimes CRISPR, sometimes other things like zinc fingers, to specifically disable or delete the gene for that CCR5 receptor on a patient's own immune cells, their T cells.
To take away the keyhole HIV uses to get in?
Exactly. Block the main entry route.
Mm.
There are active trials, like one called EBT-101 from Excision Biotherapeutics, and another from Sangamo, using those zinc fingers, testing this very strategy.
Okay, CRISPR editing, CCR5 deletion. What's the third one?
The third is called CAR T-cell therapy. This is actually a technique borrowed from cancer treatment, where it's shown incredible success.
How does that work for HIV?
You take a patient's own immune T cells, engineer them in the lab to specifically recognize and hunt down cells infected with HIV, and then you infuse these supercharged cells back into the patient's body.
So you're basically training the immune system's own cells to be expert HIV hunters.
That's a great way to put it. You're creating an army of specialized cells designed to seek and destroy the virus reservoir.
This all sounds incredibly advanced. It's amazing to hear the different angles scientists are taking, but how real is this right now? Are these just concepts in labs, or are there actual clinical trials with people happening in the U.S.?
Oh, they're definitely happening. We have active trials underway. That EBT-101 trial I mentioned from Excision Bio, that uses CRISPR to go after 3 different HIV genes, not just one.
Okay.
Then there's ADT-103T from a company called American Gene Technologies. Their approach focuses more on boosting the T cell's immunity and stopping the virus from replicating. And there were the CalImmune trials, now part of CSL Behring. They were working on modifying stem cells, the cells that create the immune system, to make them resistant to HIV right from the start.
And what are the results looking like from these early trials? Any signs of success?
The early signs are genuinely encouraging, yes. We're seeing cases where patients in these trials have had their viral loads become undetectable, sometimes for months, even years in a few instances, without needing their standard daily antiretroviral pills.
Wow, without the daily meds, that's huge.
It is potentially huge. But, and this is a really important, but these are still early days, early stage studies. These therapies are definitely not approved for general use yet by the FDA or other regulators. There's still a way to go.
Okay, right. That makes sense. So, despite all this promise, all these breakthroughs, let's unpack the flip side. What are the big roadblocks? What are the major challenges holding gene therapy back from being, the standard cure right now?
There are some significant hurdles, absolutely. First off, and it's a big one, cost. Right now, these kinds of therapies can cost upwards of $100,000 per patient, sometimes much more. That's obviously a massive barrier for widespread access.
Yeah, affordability is key. What else?
Safety is paramount. We're talking about modifying the very building blocks of life, our genes. There are always risks. Could there be off-target effects where the gene editing tool accidentally changes the wrong gene? Or could the body have an unexpected immune reaction? And we honestly don't know the very long-term effects of making permanent changes to someone's DNA. That needs years, decades even, of careful follow-up.
That's a serious consideration, the unknown long-term effects.
Definitely. Then there's scalability. These aren't simple pills you can mass produce. They're complex procedures requiring sophisticated labs, specialized equipment, highly trained personnel. Making that available globally or even widely within one country is a huge logistical challenge.
Right. It's not like setting up a standard clinic.
Not at all. And finally, there are the ethical concerns. Anytime we talk about editing the human genome, especially in ways that could be permanent, it raises deep ethical questions. Questions about what's appropriate, what the unforeseen societal consequences might be. We're still grappling with those as a society.
Cost, safety, scalability, ethics. Okay, those are definitely major hurdles to overcome. So when you weigh the incredible potential against these significant challenges, what's the outlook? Where do we stand? Are we actually close to seeing a permanent cure become available?
Well, connecting all these threads, I'd say we are certainly closer than we have ever been before. That's undeniable. But are we there yet for widespread use? No, not quite. The positive signs are clear, though. The trials are getting larger, the results seem to be improving, there's more funding flowing into this research, and public and scientific awareness is growing.
So cautious optimism.
Cautious optimism is a good way to put it. Some experts in the field, people deeply involved in this work, they genuinely believe we might see the first FDA-approved gene-based functional cures within the next, say, 5 to 10 years.
Functional cures, meaning?
Meaning the virus is suppressed down to undetectable levels long-term, potentially without needing daily medication. Even if maybe tiny traces of the virus still technically remain hidden somewhere in the body. It's not necessarily absolute eradication, but it's a state where HIV doesn't impact health or require constant treatment.
Okay, a functional cure in 5, 10 years, that would still be revolutionary.
Absolutely. But until that day arrives, and even after, we have to remember the tools we have now.
Hmm?
Routine testing is critical. Early diagnosis makes a huge difference, and ensuring everyone has access to effective antiretroviral therapy remains absolutely fundamental. Those are still our strongest weapons today.
Right. So, just to wrap up our deep dive today, the main takeaway seems to be that gene therapy, it's not science fiction anymore when it comes to HIV. It's one of the most exciting frontiers, evolving incredibly fast from theory into real potential in labs with actual patients involved.
It truly is. And maybe a final thought to leave people with something that builds on all this. Given how fast things are moving, how much more we're learning about genetics, how might these gene therapy tools, developed initially for something like HIV, eventually be applied elsewhere? Could we see them used for other complex chronic diseases? And maybe more profoundly, as we get better and better at controlling our own genetic makeup, what new ethical questions, what new dilemmas are we going to face down the road? That's something we'll all need to think about.